So my lecture tonight, it needs to begin with a confession. So this is an after-dinner lecture, and at an after-dinner lecture, you're supposed to tell jokes. But when I tell jokes in my lectures, the graduate teaching assistants come up afterwards and say, Bill, you know, that was a good lecture, but don't tell jokes. So with that apology, I'll go on and tell mainly science tonight, and maybe a little bit of a joke somewhere along the line. So I'm going to touch on three broad areas: the brain, electrical signaling, and epilepsy. And as you can all imagine, I can't tell you very much about any of them, but I want to try and tell you about one little intersection between these three large areas of neuroscience and medicine. And I appreciate very much the invitation from Nancy to be part of the molecular medicine lecture series. It's very important for us at the university to communicate as best we can the advances in medicine to the public, and I'll try and do my best at that in my lecture tonight. So even though it's after dinner, I want to follow along with a menu for tonight's lecture, and tonight's lecture has four parts, like a good Italian dinner. So we're going to start with the antipasto, and the antipasto addresses the question, how does the brain generate electrical signals to receive and process information? And this is an old subject. People have worked on this for literally centuries, and a lot has been known for as long as 50 years. And so to give you an introduction to this important and very large area of neuroscience, I've chosen a few slides from major figures in neuroscience in the past century to illustrate some of the key points. So here on this first slide, drawn from a paper by Hartline in 1934, you can see the electrical signals generated by the eye of the horseshoe crab when light is shined on the eye. And these electrical signals are recorded in the nerve that leads from the eye back to the brain of the horseshoe crab. And you can see when there's a low level of light here, the electrical signals that are generated are relatively infrequent, all of the same size, and these electrical signals bring the information back to the brain about the light falling on the eye. If the light is more intense, the electrical signals are the same size, but they come at higher frequency. And if it's even more intense, 100 times more intense, then the electrical signals generated in the eye, carried back to the brain, are at higher frequency. And this little experiment makes an important point, and that is that very often information in the nervous system and in the brain is encoded in the frequency of electrical signals that are generated. Now imagine if you could actually sit in the brain and listen to these electrical signals coming in to the visual cortex, the part of the brain that receives this information, now in a mammal. Well, that kind of experiment was done by Hubel and Wiesel in the 1950s and is illustrated on this slide. And now I'll try and speak quietly because you'll hear the electrical activity as they recorded in the brain of a cat when they moved this shining bar across the retina of the cat. Okay, so they move the shining bar here, and when they get to a specific spot, you can see that the nerve cells in the brain receive information from the eye and fire electrical signals that you're hearing through an amplifier and microphone in their experimental setup. So now they've marked the sensitive spot. Now if they move the bar, there's no more electrical signaling because these nerve cells in the brain are only responding when the light is right here and only in this pattern of the bar. So there's the sensitive spot, not sensitive here. Now they'll make the bar bigger, and you'll see even with a bigger bar of light, if it's away from the sensitive spot, no electrical signals. Then when it goes over this area, we'll bring it over there. There we go. So now they're marking the edge of the sensitive area on this side, the edge of the sensitive area on the other side. And watch what they do now. They turn the bar around, run it across the spot in the other direction. Silence. So these nerve cells in the brain only respond to a bar in the correct orientation, falling on the retina in the right place. So this is an example of the extreme specificity of electrical signaling and shows you that we represent the peripheral world in our brain in terms of electrical signals. So here, a slide from Marcus Reichle to illustrate the idea that electrical signals represent different things in different parts of the brain. So here we're using the word words. When you see words written on a page, you have electrical signals in your visual cortex here at the back of the brain. When you hear words, as in speaking, you have electrical signals here in the part of the brain that is important for hearing. When you speak words, your brain sends out electrical signals from here, another part of the brain specialized for speech. And when you think about the concept of words, electrical signals are generated here in the frontal cortex, in the decision-making and thinking part of the brain. So again, you see that all the brain does really is represented and processed in terms of electrical signals. But the brain doesn't have a battery and it doesn't have any wires. How exactly are electrical signals generated in the brain? Well, this has also been known for a long time, at least in general terms. So here, based on the work of Hodgkin and Huxley in the 1950s, is an explanation of how the brain makes electricity. And there are no electrons or wires involved, as you can imagine. So the brain uses the electrical charge on sodium atoms here and potassium atoms. And when they're charged, we call them ions. So sodium ions and potassium ions are the electrical current carriers in the brain. And when the brain makes those very brief electrical signals that I showed on the earlier slide, first sodium ions rush into the nerve cell or the nerve fiber, and they cause the rising phase of the electrical signal called the action potential. And then just a short time later, a millisecond later, that's 1/1000th of a second, sodium ions stop going into the cell and potassium ions rush out of the cell and bring the electrical signal back to the baseline. So these two changes in ion movement, sodium into the cell and potassium out of the cell, on the millisecond time scale, generates those electrical signals that we saw on the previous slides. And in fact, we know now that ion channels, proteins in the cell membrane that transport these sodium and potassium ions across the cell, are really what's responsible for these electrical signals in the brain. So this brings us to the next course of our Italian dinner here, the primi or pasta course. Now, I had an Italian dinner tonight too with the graduate students and undergraduates. We had pizza, and because I had a grant to do, I arrived late, so I had cold pizza. So your Italian dinner is better than mine tonight because you get pasta, and hopefully it'll be still warm when we go through this part of the talk. The question that I was interested in when I moved to the University of Washington and set up my lab is the one illustrated here on the slide. What's the molecular machine that generates electrical signals in the brain? Just how do the sodium and potassium ions move across the membrane? What is the membrane protein molecule that does that job? And at that time, we knew that many different neurotoxins made by scorpions and snakes and other venomous animals paralyze their prey by blocking electrical signals, by preventing prey from moving because the brain couldn't tell the arm or the leg or whatever to move. And so we used toxins from those venomous animals, for example, the scorpion illustrated here, as molecular fishing hooks. And we fished out the protein molecules in the brain that form the sodium channel. And we found 3 protein molecules in the brain that make up sodium channels. We call them the alpha, beta 1, and beta 2 subunits. And the alpha subunit is quite a large protein. Beta subunits are much smaller proteins. And you can see here our drawing at the time of how we thought the alpha subunit was organized across the nerve membrane to make a pore through the membrane through which the sodium ions move. And we also knew at that time that the alpha subunit contained the voltage-dependent gating apparatus of the sodium channel, the part of the channel that senses the electrical potential across the nerve membrane and tells the pore to open and close and let sodium ions in. Now, we knew this protein we purified was really a sodium channel because we could actually record electrical signals from single purified sodium channel molecules. The size of these electrical signals is measured in picoamperes. That's 1/millionth of 1/millionth of an ampere. So a very small amount of electrical current, but still measurable with sensitive techniques. Now, this work was done quite a long time ago, and you can tell how long it might have been from the next picture. Notice the color and amount of hair on the speaker. This is a lecture I gave on this subject in Kyoto at the International Congress of Pharmacology in 1984. So over the years, while my hair has been getting thinner and grayer, we've had a lot of fun learning more about exactly how the sodium channel works. And now, especially for the public audience, I want to take a couple of minutes to talk about proteins and then come back to the theme of the sodium channel protein. So proteins are made of building blocks called amino acids. There are 20 of them. And those building blocks are strung together in long strings and then folded up to make the structure, the three-dimensional structure of a sodium channel or another protein molecule, as illustrated on the previous slide. So for a membrane protein, those building blocks are stitched together like this and often fold back and forth across the membrane to make a three-dimensional structure of a protein in the membrane. And of course, the sodium channel is that kind of membrane protein. So now we jump back to the sodium channel and see how it is folded in 2 dimensions back and forth across the nerve membrane. So the sodium channel alpha subunit, illustrated here, is composed of 2,000 amino acid residues. So I haven't tried to draw each one as an individual block. But those 2,000 amino acids are threaded back and forth across the membrane 24 times, as you see here, still the world's record for transmembrane segments in a protein. But its organization is a little easier to understand if you realize that it consists of 4 repeated domains, each containing 6 transmembrane segments. And I'll call these S1 through S6 for segment 1 through segment 6 in the talk. And the slide illustrates that the pore in the sodium channel, the part of the channel that actually conducts sodium into the cell, is formed by the S5 and S6 transmembrane segments that are highlighted in green here. The other part of the sodium channel that's crucial is the voltage sensing module. The voltage sensing module is made up of the S1 through S4 segments, and again, there's a replica in each of the 4 domains. Of special importance in the voltage sensing module is the S4 segment that contains positively charged amino acid residues. You can see the little plus signs symbolizing that here. Those positive charges arrayed across the membrane sense the electric field in the membrane because they are a charge in an electric field and the field exerts a force on them. And it is these positive charges here, often called the gating charges of the sodium channel, that sense the electric field and tell the pore to open up and let sodium ions through. So I want to go on now from this 2-dimensional view, the exploded view, if you will, of the sodium channel, and show you how we think it looks in 3 dimensions in a nerve cell, and then how we think it works in 3 dimensions in a nerve cell. So the first step on this process depends on work from Rod McKinnon's lab at Rockefeller University, who was able, for the first time, to determine the 3-dimensional structure of a voltage-gated ion channel protein related to sodium channels. He did that by X-ray crystallography. And we've drawn this image of the sodium channel based on his work. And it shows 2 of the 4 domains of a sodium channel surrounding the central pore through which the sodium ions move. On the left, you can see 1 of the domains. On the right, 1 of the 4 domains. And then there's a 3rd domain behind the plane of the slide, and a 4th domain in front of the plane of the slide that I have left out for clarity. So the pore through which the ions move in the center of the molecule is formed here, and 2 ions are shown in the pore as they would be if they were being transported through it. And as I mentioned before, the pore is formed by the S5 and S6 segments in each of the 4 domains of the channel. The voltage sensing module is here. It contains 4 transmembrane segments in a bundle, and the S4 segment, here in green, is the one that has those crucial gating charges located within it. So this work from the McKinnon lab then shows us for the first time how the parts of the sodium channel are arranged in a 3-dimensional context. Now, to understand how the sodium channel works, we need to know the structure of the sodium channel in each of its active and inactive states. So this is the structure of the sodium channel in its active state. The pore is open. These little ions could move through if they were able in our still picture here. With David Baker in the Department of Biochemistry at the University of Washington, we used molecular modeling methods, and this structure of the activated state of the sodium channel to create a working model of the sodium channel, which moves from the resting state to the activated state when I press a button in a couple of minutes. But before I click to make it move, let me again highlight the key features and tell you what to expect. So here, the center is the pore-forming module. In this case, the pore-forming module from each of the 4 domains of the channel. So the sodium ions would move right through the middle here, and you'll see that happen in a slide or 2. Then the voltage sensing module here, the key voltage, the key gating charges, which actually feel the electric field and cause the channel to change from the resting or closed to the activated or open state, and then this key linker here that links the voltage sensing module to the pore forming module. And as you'll see in just a moment, when we activate the channel, this S4 segment moves outward. Each of these positively charged gating charges moves past this little red ball, which signifies a negative charge in the middle of this voltage sensing module. It pulls on this linker, and the linker pulls open the pore, and there are no sodium ions here, but if there were, they would go right through. So here's the motion, and you can see how that works. I'll run it again. So watch how this moves out, pulls here, opens here, makes an electrical signal. So this is how we think the sodium channel works at really the detailed biophysical level to respond to changes in voltage in the nerve cell membrane and to make an electrical signal or an action potential. Now, as I mentioned on one of the early slides, electrical signals are always brief. They have to be brief because remember that information is encoded in the frequency of electrical signals. So you have to be able to generate them one after the other. If you just turn the sodium channel on and left it on, that would be one electrical signal and the brain would stop. So the sodium channel has a second process, we call it a voltage-dependent gating process, that turns it off within 1 millisecond after it's turned on. And that makes sure that every electrical signal is brief, as you need to have in order to encode information in the brain. And there's a specialized part of the sodium channel that does that job, which I haven't mentioned before. It's this little part of the channel right here that connects the third domain and the fourth domain. And we call this part of the sodium channel the inactivation gate because it folds over the pore and inactivates the channel, just like a gate on a garden path. And with Rachel Clevitt in the Department of Biochemistry, we've been able to determine the structure of the inactivation gate, and I won't tell you the details about it, but you can see it here. And then we have put the inactivation gate on our sodium channel model in the next slide. So now we have the full sodium channel orchestra to watch here. Voltage sensing module, pore forming module. Now we've got sodium ions. They're not green really, but they're green here. And the inactivation gate on the intracellular side. And so now when we press the button, we'll get activation of the voltage sensing module, pulling open the pore, sodium ions moving, inactivation gate closing, and that's a full electrical signal in a nerve cell, just like that. And then of course, if you're a nerve cell, you keep doing it, one after another, and generate all that train of electrical signals carrying information, for example, from the eye back to the brain. Now, the sodium channel is very important physiologically. I've shown you the important job that it does here. It's also an important target of drugs that are used in the clinic. Now, one you're very familiar with is the local anesthetic Novocaine. So when you go to the dentist injects Novocaine into the nerves under your teeth. Novocaine blocks the sodium channel, so it can't work. And then you don't know, and your brain doesn't find out about the bad things that the dentist is doing in your mouth. So this is a really important drug. We're really glad that we have local anesthetics. Other sodium channel blocking drugs are important in different conditions, and in epilepsy, some anti-epileptic drugs block sodium channels in the brain and prevent the uncontrolled electrical signaling in the brain that is the cause of epileptic seizures. And we know a lot about how the local anesthetic and the anti-epileptic drugs act on the sodium channel at the molecular level. And I'll just show you this one more piece of our sodium channel model. So here again, the kind of two-dimensional folding model of the sodium channel. The drugs, the local anesthetics and anti-epileptic drugs that act on sodium channels bind within the pore of the channel, and they bind to a receptor site that's made up of the S6 segments in 3 of the 4 domains, in domain 1, in domain 3, and in domain 4. And here we see that in higher resolution. You can see the S6 segment here represented as an alpha helix from domain 1, domain 3, domain 4, a local anesthetic molecule sitting nestled in its receptor site, blocking the pore. And here, the space-filling view of the amino acids that surround it and hold it in place. So this is a very important drug receptor site, both for the dentist and for the control of seizures. Okay, so that finishes our pasta course here, and we need to go on to the secondi, the main course of our lecture. And this is a little bit longer than the other parts of the lecture, so I guess it's fair that it turns out to be our main course. So now I want to turn from the sodium channel to epilepsy. So we've learned a little bit about how the brain uses electrical signals, a little bit more about how the sodium channel generates electrical signals, and now we'll learn about how uncontrolled electrical signaling can cause a genetic form of epilepsy and also can cause its comorbidities, that is to say, other aspects of the disease besides the seizures also caused by uncontrolled electrical signaling. So a little word about epilepsy. I imagine that you all know that epilepsy is recurrent, spontaneous, episodic seizures. So often, an individual have a single seizure, for example, after a head trauma or an infection in the central nervous system, one might have a single seizure. We don't speak of that as epilepsy. So epilepsy is recurrent, spontaneous seizures without any cause provoking them right at the time of the seizure. And these seizures in epilepsy are caused by synchronous electrical firing of all of the neurons in either an area of the brain or sometimes in the entire brain. So epilepsy really is uncontrolled electrical signaling. Usually, this is due to a localized hyperexcitability in an epileptic focus. That is one area of the brain where the neurons are generating too many electrical signals, and then it excites the neighboring areas, and soon all of the brain is involved in an epileptic seizure. So seizures can be focal when the excess electrical signaling is isolated in an area of the brain, or they can be generalized throughout the brain. Epilepsy is caused by several different kinds of insults to the brain. It can be caused by brain injury, as in an infection or after head trauma or after a brain tumor, for example. It can also be caused by genetic mutations that specify abnormalities in ion channels or other proteins in the brain. And these mutations might be inherited from the parents of the person who has epilepsy, or they might arise as new mutations, de novo, in the person who has epilepsy. So, as you can see from this description, epilepsy is a very complex disease with multiple courses and, although I haven't described it, very complex outcomes. So to try and understand it better, we have focused on a single type of epilepsy, which is a very severe but rare form of childhood epilepsy called severe myoclonic epilepsy of infancy, and I'll abbreviate it SMEI. So SMEI is caused by mutations of one of the sodium channels in the brain. It's called the NAV1.1 channel. It's one of 4 different sodium channels that are made from 4 different genes in the human brain and also in the brains of other mammals, such as the mice that I'll introduce you to in a minute. So this is a very severe epilepsy. As I mentioned, it's caused by de novo loss of function mutations with a dominant phenotype. That is to say, only one of the 2 genes that encodes the NAV1.1 channel needs to be mutated to cause this disease. It's dominant. It's a very severe childhood epilepsy and is a significant amount of premature death among children with this disease, so it's extremely severe. It usually begins with febrile seizures, usually before 1 year of age, and then it's followed by spontaneous seizures that become progressively more severe and are intractable in the sense that they're not well treated by anti-epileptic drugs. This epilepsy has several serious comorbidities, including ataxia and spasticity, cognitive impairment, and visual and circadian rhythm disturbances. The cognitive impairment is so severe that children that survive the seizure phase of this disease and become teenagers have IQs in the range of 40 and need institutional care for all of their life. So, fortunately, this is a very rare epilepsy, but as I've described to you, a very severe one. Because this is a genetic epilepsy, we can recreate the epilepsy in a mouse. And we've done that by introducing a mutation into the corresponding NAV1.1 channel gene in the mouse. And the heterozygous mice, and remember now, the humans with this disease are heterozygotes. They have a good gene and a defective gene. So the heterozygous mice are viable and fertile. They have thermally induced seizures after postnatal day 20, and I'll show you this. And this is analogous to the febrile seizures in human infants. After they are susceptible to thermally induced seizures, they have spontaneous, generalized tonic-clonic seizures or grand mal seizures after postnatal day 21. Many mice die prematurely, and they have all of the comorbidities of the human disease, including ataxia and cognitive deficit. So we're very fortunate because often when one introduces a human mutation into mice, it doesn't mimic the human disease process. Here, it's almost an exact mimic, so we feel fortunate in that regard. So the first two questions that we tried to answer with this mouse model are indicated on the bottom of the slide, and the first question really is a paradox. Why does loss of sodium channel function cause hyperexcitability and epilepsy? From what I've told you so far, the sodium channel drives the electrical signaling in the brain, yet here, loss of sodium channel function is causing uncontrolled electrical signaling. So can we figure out why that is in the mouse? And then secondly, what causes the comorbidities that accompany this epilepsy? And I'll focus on ataxia in mouse SMEI. Now, I have to digress for a couple of minutes to go back to telling you how the brain works before I can really explain the next series of experiments on this epilepsy. So, there are really two broad classes of neurons in the brain with respect to electrical signaling. There are excitatory neurons. When they generate an electrical signal and pass it along to the neighboring nerve cells, they excite those neighboring nerve cells. And then, on the other hand, there are inhibitory neurons in the brain. And they're very important. They actually control the electrical signaling of the excitatory neurons. So when the inhibitory neurons generate an electrical signal, the neurons that they contact are inhibited. They're inhibited by a neurotransmitter called gamma-aminobutyric acid, or GABA, which, when it is released onto a neighboring neuron, inhibits the electrical signaling of that neuron. So the importance of this can be illustrated by a simple example in the reflex arc that controls our muscle from the spinal cord. So when you extend your arm, the motor neurons in your spinal cord tell your muscles to extend, right? Now, when you retract your arm, the motor neurons in your spinal cord tell your muscle to flex or retract, bring your arm back. If you have both of these neurons doing this at the same time, then you have totally uncoordinated movement. That's prevented by an inhibitory neuron, which tells this flexor motor neuron not to fire when this extensor motor neuron is firing. So you get smooth extension of your arm without reflex return as long as these inhibitory neurons are working. So you can see how important inhibitory control is in this simple system. And of course, the brain has billions of excitatory and inhibitory neurons working with this same principle. So this raises the question, could impaired excitability of inhibitory neurons cause the hyperexcitability in SMEI? So this is really the yin and yang of electrical signaling. There's a balance between excitatory and inhibitory electrical signaling in the normal brain. And we're going to ask whether that balance is altered in SMEI. So we asked that question first in an area of the brain deep under the cerebral cortex called the hippocampus. It's located here in this image of a human brain, or here in higher magnification. And it's known that this area of the brain is very important in epilepsy. And like all the other areas of the brain, this area has excitatory and inhibitory neurons. So our experiment is to dissociate those neurons, identify them, and then use electrical recording techniques to ask, is the electrical signaling in the excitatory neurons or the inhibitory neurons preferentially affected by our SMEI mutation? So for this part of the talk, I guess this is the key experiment, and so I'll go through it kind of slowly. So here we're measuring, using an electrical technique, the electrical current, we call it the sodium current, that's moving through the sodium channels in these nerve cells. The left side of the slide here shows the experiment for the excitatory neurons. They're called pyramidal cells in the hippocampus. And the plot shows the size of the sodium current, which increases in the downward direction. It's a negative quantity, so it's plotted downward. So as we stimulate the nerve cell more and more and more and more and more, the sodium current gets bigger, 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 and at this potential, about minus 10 millivolts, you see a maximum sodium current, and then it gets smaller. So the key experiment here is to compare the wild-type mouse, plus plus, both good genes, the heterozygous mouse, plus minus, one bad gene, and now the knockout mouse, two bad genes. Now this is not the analogy of the human disease. The analogy of the human disease is the heterozygote, but we can also study the homozygous knockout mouse. And the key result here is that for the excitatory neurons, nothing's happening. You take away this sodium channel and they don't care because they're using the other sodium channel genes to drive their electrical signaling. Now the story is completely different on the other side of the slide. Here, the hippocampal interneurons, the inhibitory neurons of the hippocampus, we take away one of their NaV1.1 genes and they lose half their sodium current. And if we take away the second one, they lose another 25%. So we're seeing a selective impairment of the electrical signaling of the inhibitory neurons with no impairment of the excitatory neurons. So one would predict that there would be uncontrolled electrical signaling in the hippocampus of these mice and of human patients with this genetic lesion. We can also measure the electrical signaling in the inhibitory neurons in the same way that I've shown you in previous slides by measuring action potentials. And this slide shows you that for the wild-type mice, plus plus, as we stimulate the neurons more and more and more and more with increasing amounts of stimulating current, they can fire action potentials happily for this whole period of time at increasing frequency, and notice the frequency goes up as we stimulate more, but they're always able to fire continuously. So this is their normal behavior of these inhibitory neurons. So if we take away one of the two sodium channel genes, look what happens. These neurons can no longer keep up with the action potential firing that they need to do in order to control the excitatory neurons in this part of the brain. And if we take away both genes, of course, their firing is dramatically impaired even more. So we think then the answer to our paradox is that hyperexcitability and epilepsy is caused in this mouse model, and we imagine in the human disease, by disinhibition, by the failure of firing of the inhibitory neurons, letting the excitatory neurons fire in an uncontrolled way. Now, what about the comorbidities, the other aspects of this disease which are so important to patients? So the comorbidity we have studied first in mice is ataxia. And so the first question is, are the mice ataxic? We have to measure and show that actually they are ataxic. And so this slide shows you a very simple test of mouse ataxia. It's called a footprint test. We get the mice to walk through a plexiglass tunnel with a digital camera underneath, and we record the position of their feet on each step. And a wild-type mouse is very precise. You probably hadn't noticed this in the mice around your house. Each step, they put their hind paw exactly where their forepaw had been on the previous step. And you can see that here. The white circles and the dark circles illustrate the position of the hind and forepaw on each step. And you can see they pretty much overlap for the wild-type mouse, but they don't overlap for the heterozygous mice. So the heterozygous mice aren't able to walk normally. They can't put their hind foot where it's supposed to be when they walk. And you can do statistics and measure those distances down here, and you can see there's a much bigger distance on average between the positions of the front and rear paws of the mutant mice compared to the wild-type mice. And they're also imprecise about where they put their feet. This error bar here shows you the variability in placement of their foot, which is much greater than for the wild-type mice. So this is kind of a drunken sailor walk for mice. And so the hets, the mutant mice, are indeed ataxic. So why might they be ataxic? Well, there's a part of the brain very important in controlling movement, the cerebellum. It's located here, below the cerebral cortex and forebrain, and here in the hindbrain, below the hindbrain. So the cerebellum is crucial for controlling movement, and within the cerebellum, a crucial set of neurons, the Purkinje neurons, are inhibitory neurons. So could it be that these neurons are also losing their ability to fire electrical signals and control the movements of the mice in the proper way? So this is an illustration of the Purkinje neurons in the cerebellum. You can see they're rather beautiful nerve cells. They all line up in a row, and they have these lovely dendrites that project up from them. So we did the same experiment. We dissociated those neurons out of the cerebellum, and we studied them electrically, and now you're all good electrophysiologists, and you can see immediately. The wild-type cerebellar neurons fire action potentials at high frequency, the heterozygotes at intermediate frequency, and the knockouts at much slower frequency. And this failure to fire at the normal rate is likely the reason for ataxia in our mice and in human children with this disease. So we think then that not only the epilepsy is caused by the failure of firing of GABAergic neurons, but also the comorbidities in this disease are caused by the same failure, but in different classes of neurons that have different jobs to do. And we illustrate that here with the neurons in the cerebellum and with ataxia. Okay, so we come to the last course of our Italian dinner here, dolce, the dessert. Now, dessert should be short, right? Because after all, we don't want to eat too much. So this is a short course, but it's an important topic. Can we learn anything from our mouse model to tell us how we can improve therapy of this genetic epilepsy? So first, we need to have a measure, a quantitative measure of the effect of the drugs, anti-epileptic drugs, on epilepsy in our mice. And so we've used this kind of experiment which tries to mimic febrile seizures in humans. And you recall that SMEI usually begins with a child having a febrile seizure, which then leads to a more severe epilepsy syndrome. So our experiment is to put a mouse in a chamber here with a rectal thermometer to take its temperature. And we record the temperature, and then we have a heat lamp which allows us to raise the temperature of the mouse, and we have a feedback circuit so that we can raise it slowly and steadily as it would be raised during a fever in a disease in humans or in mice. We record behavior of the mouse on a video recorder, and we measure from outside the cranium, but with electrodes on the skull. We measure the electroencephalogram, the EEG, which is a measure of brain waves and brain electrical activity. Then we do this experiment. So here, an example of a wild-type mouse. So we've raised its temperature steadily up to 44 degrees centigrade. This is much higher than a fever. The highest temperature for fever in humans is about 40 degrees centigrade. And then we cool the mouse down. Wild-type mice don't have seizures. They're perfectly happy after you cool them down. And so over this 5-minute period of time, wild-type mice never have seizures. On the other hand, in our heterozygous knockout mice, once the temperature gets above about 40 degrees, so just at the limit of fever in humans, the mice have seizures. And in a 5-minute experiment, they typically have 1 or 2 seizures per mouse, whereas the wild types have none. So we can, in this way, generate seizures when we want, so to speak, in our mice, and we can ask if drugs can block them. So the right side of the slide shows the electrical recording of the seizure. So here, the EEG of the mouse when it's just carrying on about its cage in normal business. You can see there's some electrical activity, but it's not dramatic. As we begin to warm the mouse, the first sign of trouble are these spikes here. You can see these large electrical spikes in the EEG, and each one of these is associated with a myoclonic seizure. Now, a myoclonic seizure is a quick jerk of the arm or the head or the leg, or in the case of the mouse, the tail. You can see that in the behavioral trace that goes along with these electrical records. Then as we warm the mouse further, eventually the myoclonic seizures become more frequent, and then the mouse has a tonic-clonic seizure or grand mal seizure with much electrical activity all over the brain, as you can see from these records. Then as we bring the temperature back down, the mouse has some more myoclonic seizures and eventually has a very quiet EEG thereafter. So we can measure seizures in the mouse. Can we prevent them? So again, I have to go back to telling you a little bit about the brain. This is the last bit of brain science for tonight. So I haven't said very much about synapses in the brain. So the synapse in the brain is the place where one nerve cell contacts its neighbor and sends either an excitatory or an inhibitory message to the neighboring nerve cell. And so this is a drawing of an inhibitory neuron and an inhibitory synapse. So the electrical signal would start way over here in the cell body, over in the next room, come all the way down the nerve fiber. The electrical signal, when it reaches the nerve terminal, causes the release of the neurotransmitter GABA from these little packets, little vesicles. The GABA diffuses across to the next nerve cell where it encounters a GABA receptor. And the GABA receptor binds the GABA and tells this nerve cell, quiet down, don't fire any electrical signals, I've got inhibitory word from upline. And so then this cell is inhibited. GABA in the synaptic cleft that doesn't bind to the receptor is brought back into the nerve terminal by a GABA transporter right here that cleans up the leftover GABA. So now in our situation, the synapse is not getting the electrical signal, right? It's not getting the word to release GABA, and so GABA isn't being released, or it's being released much less. So somehow we want to tune up the function of this synapse to work with less GABA. And we've done that by a combination of drug treatments. So first, we use a drug called a benzodiazepine. The drug Valium is a member of this drug class. Probably you've heard of Valium. It makes the GABA receptor more responsive to GABA. And we use the drug tiagabine here. It blocks this uptake of GABA so that GABA that is released stays longer in the synapse between the cells, can bind the receptor, and the receptor response is enhanced. So we hope with this two-pronged treatment we can improve the situation for our mice and eventually perhaps for patients. So here's some of our early data on mice. This is another one of those thermal induction experiments where we're warming the mouse to generate seizures, and in control, that is with no drug here, we're warming the mouse, and you can see the body temperature of the mouse here on the x-axis. The y-axis measures the number of mice that are without seizures, and so they all start without seizures, so we start at 100%. As we warm the control mice, more and more of them have seizures, and eventually they all have seizures by 39 degrees, so in the range of fever for humans. When we add this combination of drugs, we greatly increase the temperature that we have to raise the mouse to before they have seizures. And we get up to the range, the highest range of human fever with this drug combination. So this is a very positive drug effect. We're preventing the seizures in the normal temperature range for mice and for humans. So we think this is very encouraging. And importantly, as I mentioned, humans with this disease have significant premature death, and our mice also suffer premature death with this disease. So here we're measuring the percent of mice that remain alive. Of course, at the beginning that's 100%, as a function of their age. So we're not doing anything to them except letting them grow and go about their business. When they start having seizures at postnatal day 21, many of the mice die. And by postnatal day 120, about 70% of the mice have died. with our combination of clonazepam plus tiagabine, we can greatly reduce the deaths of the mice, not completely, but we can reduce it by a factor of 2. So again, this is very encouraging. It lets us think that this idea of enhancing inhibitory neurotransmission with combinations of drugs that are not now used in human therapy might be very useful, first in curing the mice, and then most importantly, perhaps in helping the children who have this disease. So with that now, I'd like to go to my very last slide and close. And as you can imagine, I haven't done most of the work that's been described tonight, not even most of the work done in my own lab. And I've summarized work from more than 30 years here at the University of Washington. So first, I have to thank many unnamed graduate students and postdocs who've contributed to that long series of experiments on sodium channels and on epilepsy. Now on the slide, I've noted the names of people more recently in the lab who've contributed the experiments that I showed in more detail. And because the hour is late, I won't read their names, but you can do that here. The 4 people who were critical for the sodium channel experiments, the people who were critical for the epileptic mouse experiments. I want to note particularly my faculty colleagues in pharmacology, Ruth Westenbrook and Todd Scheuer, who've been collaborators over more than 20 years of the 30 years that I've been here, and we're involved in all of these studies. UW is a great place to do research because every corridor you go down, there's another potential collaborator, and here are several that have been important for our work over the years. I thank my colleague Bertil Hille in physiology, who also studies ion channels, for some of the introductory slides. And special thanks to the National Institutes of Health, NIH, and the McKnight Foundation for generous support for our research. And finally, thanks again so much to you for coming out on a nasty night to learn a little bit about the brain and epilepsy.
So you mentioned earlier in your talk that there were 4 different types of sodium channels found in the brain, of which NAV 1.1 is one. How much homology is there among those 4 types of sodium channels, and does it present a difficulty in designing new drugs to treat epilepsy?
The sodium channels are very highly conserved, as you might expect. In humans and other mammals, there are a total of 10 sodium channel genes. As I mentioned, 4 of them are expressed in the brain, but there are 6 that are expressed elsewhere. Most of the drugs that are used that block sodium channels, including the ones that I mentioned, are not selective among the different sodium channel types. And this is a real deficit in our modern pharmacology of sodium channels, and many drug companies are working hard using the kinds of genetic and structural information that I've shown you tonight to try and design drugs that would be specific for individual sodium channels. And these would make better local anesthetics, they would make better anti-epileptic drugs. Companies are interested in sodium channel blocking drugs for pain because, of course, pain signals come by way of electrical signals as well, and in drugs used for treatment of arrhythmias in the heart. So it would be a big advance if we could make drugs that would be selective for each of the 10 sodium channels, but that's not possible now. It's a good question.